All right, now we're going to go over Chapter 4, Biology of the Cell. So cells are the functional units of the body. In fact, they are the smallest functional living units there are. All body processes depend on cellular activity. So your cells have to be living in order to have cellular activity. Cytology is the study of cells. Most cells must be viewed with a microscope because they are so small. So most cells in the human body require a microscope to be viewed. As you can see on this chart over here, you're looking at the micron range for most cells. When you get down to the nanometer range, you're talking more about viruses, proteins, and then even smaller than that, you're talking about very small molecules like amino acids or even atoms. Then you come up here above the micrometer range, you've got the human oocyte, which is the human egg cell. Some of those can be viewed with the naked eye. And then further up, you get to almost a meter, and you'll see some muscle and nerve cells are this large. Specifically the sartorius muscle, which is the longest muscle in your body, and neurons of the sciatic nerve, which is the long nerve that runs down the back of your leg to your foot. So in this chapter, we'll talk pretty extensively about the structure of a cell. We'll also talk about the function of all of these different structures that make up a cell. So we've got, going from outside in, we've got the plasma membrane on the outside. And you can have modifications to the plasma membrane. So you can have microvilli, you can have cilia, and you can have flagella. You have the cytoplasm, which is the substance inside of the cell, and it contains several different organelles. It contains, for instance, the cytoskeleton, which are a bunch of small fibers that give the cell shape and structure. It contains a centrosome, which helps in mitosis, in dividing the cell. And it contains organelles like the proteasome, ribosomes, lysosome, peroxisomes, mitochondria. And then you've got, going further in, you've got the Golgi apparatus. You have the smooth endoplasmic reticulum, the rough endoplasmic reticulum surrounding the nucleus, and here's the nucleus with a nucleolus inside of it. And we'll talk in detail about what each of these structures do. These two charts I left in your PowerPoint. I think that they will be useful study guides for you. So they're in your book, and I just wanted you to take a glance at them now. And when it comes time to study, you probably will want to reference these, table 4.1. All right, so just some general functions that cells perform. They maintain integrity and the shape of a cell. This depends on the plasma membrane and the internal contents of the cell. They obtain nutrients and form chemical building blocks. That is, they harvest energy to survive. They dispose of wastes, and this avoids accumulations that could disrupt cellular activities. And then they undergo cell division. In cell division, they make more cells of the same type, and this helps to maintain the tissue by providing cells for new growth and replacing dead cells. But it's important to remember that not all cells undergo mitosis. Okay, so first we're going to talk about the plasma membrane. This is a phospholipid bilayer, and it forms a barrier where the cytosol remains inside of the cell, and the interstitial fluid is on the outside of the cell. It regulates the movement of most substances in and out of the cell, but for reasons we'll see in a minute, small and non-polar substances are still able to cross it. It's composed of lipids, especially phospholipids, proteins, and carbohydrates. So here's a diagram showing you the structure and functions of the plasma membrane. Right now it acts as a physical barrier. It establishes a flexible boundary, protects cellular contents, and supports cell structure. The phospholipid bilayer separates substances inside and outside the cell. And what we mean when we say phospholipid bilayer is you can see a phospholipid here with its hydrophilic head and its hydrophobic tails, and it orients its head out towards the liquid. In this case, this is the interstitial fluid. And several phospholipids line up to do the same. And on the other side, the same thing's happening where the hydrophobic tails are pointed in towards each other, and all together this is called a lipid bilayer, phospholipid bilayer. It's selectively permeable. It regulates entry and exit of ions, nutrients, and waste molecules through the plasma membrane. And you'll see when we say selectively permeable, we mean most substances that need to move in and out of the cell are going to have to do so through channels or protein carriers, things of that nature. So here's an integral protein, which means it spans the entire length of the plasma membrane. And this looks like this could potentially be an ion channel or something like that, that when activated would allow certain ions through. That also has electrochemical gradients. So the plasma membrane establishes and maintains an electrical charge difference across the plasma membrane, and we'll talk about that in a little bit. And then, very importantly, it participates in communication, where it contains receptors that recognize and respond to molecular signals. So an example here would be this carbohydrate attached to this protein. These carbohydrates play a role in cell signaling and cell identification, and we'll get to that in a little while. So membrane proteins, these make up about half of the membrane's weight. They can float and move in the fluid lipid bilayer, and they perform most of the membrane's functions. There's two structural categories. So integral, we already mentioned, these are proteins that span the entire membrane. And then there are peripheral, which are found on the internal or the external surface of the membrane. So a peripheral protein, which doesn't look like there's here's an example of a peripheral protein. And you can see it only spans the inside of the cell. Then we can talk about membrane proteins in terms of functional categories. So there are transport proteins that regulate movement of substances across the membrane. For example, channels, carriers, and pumps. And pumps can either be symporters or antiporters, and we'll talk about all this in a little bit. Cell surface receptors. These bind molecules called ligands, for example, neurotransmitters that bind to their receptors. Identity markers not only allow the cell to communicate to other cells that they belong to the body, but it also helps distinguish healthy cells from cells that need to be destroyed. You've got enzymes. These can be attached to either internal or external surface of the membrane, and they catalyze chemical reactions. Some proteins can be anchoring sites. These secure the cytoskeleton to the plasma membrane. And then you've got cell adhesion proteins, which perform cell-to-cell attachments. And you can see an example of that right here, where one protein kind of hooks into another protein on another cell's plasma membrane. Here's pictures of all the categories we just talked about. So a transport protein might let certain molecules through and not others. A receptor has a special site, binding site, for a ligand to bind to it, and it activates the receptor, and a signal is essentially communicated to the cell. You can have an identity marker, and here's that carbohydrate group that we looked at earlier attached to a protein. And this can tell other cells that this cell is part of the body. And it can also tell other cells that this cell is healthy or it needs to be destroyed. You have enzymes, which you've learned about in detail already. You have anchoring sites. This would be an example of a protein, an integral protein, anchored to the cytoskeleton. And then we already mentioned cell adhesion protein, where one protein on one cell hooks into another protein on another cell. So this is a very useful flow chart going over the different membrane transport methods that the cell uses. For example, the first division here is between passive processes and active processes. Passive processes require no energy from the cell, and this is where substances move down their concentration gradient, right? So areas from high concentration to low concentration. Whereas active processes do require cellular energy and involves either the movement of a substance up its concentration gradient, right, from areas of low concentration to high concentration, or involves the formation or loss of a vesicle. So let's go down the passive processes pathway first, right? So remember, this is no expenditure of cellular energy, and substances are moving down their concentration gradient. So you can either have osmosis, which is the movement of water across selectively permeable membrane, right? So this will be where water moves down its concentration gradient from areas of high concentration of water to areas of low concentration of water. Or you've got diffusion, which is the movement of solutes. So osmosis is movement of water, diffusion is movement of solutes. Going further down diffusion, you can have simple diffusion. This is where no transport protein is required. The molecules are usually small and nonpolar and can simply diffuse right across the plasma membrane. Or you can have facilitated diffusion. This is where a transport protein is required. And you can have channel-mediated facilitated diffusion, where an ion moves through a channel, or you have carrier-mediated facilitated diffusion, where a small polar molecule is moved by a carrier protein. Going down the active processes pathway, this requires expenditure of cellular energy. And again, we said it involves either the movement of a substance up its concentration gradient or the formation or loss of a vesicle. So let's talk about active transport first. This is where an ion or small molecule is moved against the concentration gradient. So from areas of low concentration up to areas of high concentration. So you have primary active transport. This is where the energy source is from ATP. Where you have secondary active transport, and this is where the energy source is from movement of another substance. And this is where your symports and your antiports come into play. In a symport, two substances are moved in the same direction across the membrane. In an antiport, the two substances are moved in opposite directions across the membrane. And I'll show you what we mean here in just a little bit. Back to the top, active processes. Let's talk about vesicular transport. This involves a vesicle forming. So you can either have exocytosis, where vesicular contents are released from the cell, or you can have endocytosis, where material is brought into the cell as a vesicle is formed. And among endocytosis, you can have phagocytosis. This is cellular eating, where the cellular brings in particles inside of a vesicle. You can have pinocytosis. This is cellular drinking, where the cell brings in water and other liquids into the cell in the form of a vesicle. Or you could have receptor-mediated endocytosis, where a receptor is required. And we were going to talk about every single one of these in detail. Okay, so membrane transport is the process of moving substances across the plasma membrane. We'll talk here about passive processes of transport first. Remember, these do not require energy and instead depend on substances moving down their concentration gradient. There's two types. There's diffusion, which is the movement of solutes, and then there's osmosis, which is the movement of water. And then there's active processes of transport, which require energy, and this will be moving substances against their concentration gradient. And there's two types of active transport. There's active processes. There's active transport, and then there's vesicular transport. So first, let's talk about passive processes and diffusion. So diffusion is a movement of a substance from greater concentration to an area of lesser concentration. And if left unopposed, diffusion continues until the substance reaches equilibrium, where the molecules are evenly distributed throughout a given area. The rate of diffusion depends on two things. One, the steepness of the concentration gradient, that is the difference in concentration between the two areas. And a steeper gradient causes a faster rate of diffusion. And number two, temperature. There's a faster rate of diffusion with higher temperature. There's simple diffusion. This does not require membrane proteins and are not regulated by the cell. This is where small and non-polar solutes like O2 and CO2, some fatty acids, and alcohol or ethanol can move directly across the plasma membrane. But remember, this movement depends on a concentration gradient. So here it shows oxygen moving from the interstitial fluid straight through the plasma membrane into the cytosol, where there's a lower concentration of oxygen. On the other hand, you have a high concentration of carbon dioxide here, and it slips through the plasma membrane to where there's a lower concentration of carbon dioxide in the interstitial fluid. Opposed to simple diffusion, there's facilitated diffusion. This is the transport of charged or polar molecules is facilitated by membrane proteins. There's two types of facilitated diffusion. There's channel-mediated diffusion and carrier-mediated diffusion. In channel-mediated diffusion, small ions are transported through specific channels. And there's two kinds of channels to be familiar with. There's leak channels, which are always open. So ions are always kind of leaking out of the cell or leaking into the cell through these leak channels. And then there's gated channels, which have a physical gate that opens in response to some sort of stimulus that allows ions to cross. In carrier-mediated diffusion, this is where small polar molecules are assisted across the membrane by a carrier protein, and this moves substances down their concentration gradient from high to low. All right, so again, because this is diffusion, even though it's facilitated diffusion, remember that solutes are going to be moving from areas of high concentration to areas of low concentration. Here you can see high concentration of potassium leaking out through the potassium leak channel toward the area of low concentration. Here you have carrier-mediated diffusion. You've got glucose in the interstitial fluid. It binds to the protein. The protein changes shape and allows glucose to go inside the cell. But remember, there's a higher concentration of glucose outside and a lower concentration of glucose inside, and that's what allows this facilitated diffusion to take place. Osmosis is the movement of water. It's simple diffusion of H2O across a selectively permeable membrane. Water moves from high water concentrations to low water concentrations until equilibrium is reached. And water can move in two ways. It can slip between the molecules of the phospholipid bilayer, or it can move through integral protein water channels, which are called aquaporins. So remember that water moves down its concentration gradient. So it moves from a solution, say, of 1% solutes into a solution of, say, 3% solutes, right? Because if it's 3% solutes, that means it's a lower water concentration than the solution with 1% solutes. And it's going to move until it reaches equilibrium, where there's an equal concentration of water inside and outside of the cell. So on this graphic here, you can see lots of water on the right side of the plasma membrane compared to the left side, which does not have as many water molecules. So water is going to want to move from right to left here, or from the cytosol out to the interstitial fluid. It's going to move through aquaporins, which are those special proteins embedded in the plasma membrane. And it can also slip through the phospholipids in the lipid bilayer. And it's going to do this until it reaches about an equal concentration on both sides of the membrane. Alright, it's important to know that there are two types of solutes. There are permeable solutes that pass through the lipid bilayer. These are small and non-polar, such as oxygen, carbon dioxide, and urea. You're going to see this as the three big ones that come up: oxygen, carbon dioxide, and urea. Then you have non-permeable solutes. These are prevented from passing through the bilayer. They are charged, polar, or very large solutes, such as ions, glucose, and proteins. This is going to create differences in the solute concentration across the plasma membrane. Or, in other words, there's a solute concentration difference between the cytosol and the interstitial fluid. And the interstitial fluid is just the fluid in between the cells. So here's an example. You have, in this U-shaped container, you have a semi-permeable membrane, and on the right side over here you have a lot of water molecules and not as many solutes, and on the left side you have not as many water molecules and a lot more solutes. So what's going to happen is water is going to pass through the semi-permeable membrane until there's about an equal concentration of solutes on both sides. Osmosis and tonicity. So the cell gains or loses water with osmosis, and this causes a change in the cell volume and the osmotic pressure. And osmotic pressure is just the pressure that water is exerting. Tonicity is the ability of a solution to change the volume or pressure of a cell by osmosis. So here you have an isotonic solution, iso meaning same, and the interstitial fluid is the same concentration as the cytosol, and they give you a red blood cell here as an example. So there's no net movement of water. And you'll see the red blood cells take on their normal biconcave disc shape. However, in a hypotonic solution, the interstitial fluid is less concentrated than the cytosol, so water is going to move from areas of high water concentration to areas of low water concentration. So it's going to move from the interstitial fluid into the red blood cell. And that's going to swell the cell up. And this is what that would look like. You see they've kind of lost their biconcavity. In a hypertonic solution, however, the interstitial fluid is more concentrated than the cytosol, so water is going to leave the cell, and this is going to shrink the cell, and this is called crenation. And it deforms the red blood cell and can interfere with its ability to function. Over here in the hypotonic solution, if the concentration difference is great enough and enough water enters the cell, these cells can swell to the point that they lyse, or break open. This is a little video for you to watch. You can copy the link going over diffusion, osmosis, and facilitated diffusion. Okay, now let's talk about active processes. So there's active transport, which is the movement of solute against its concentration gradient from low concentration to high concentration. Remember, this requires energy and it requires transport proteins. The source of energy determines whether the movement is primary or secondary. So there's primary active transport, where the energy is ATP. And then there's secondary active transport, where energy is from the transport of another substance. Primary active transport uses energy directly from the breakdown of ATP, and this changes the protein shape and moves substances across the membrane. Two examples to be familiar with are the ion pumps and then specifically sodium-potassium pumps. Ion pumps are protein pumps that move ions across the membrane. They are required to maintain internal concentrations of ions. For example, there are calcium pumps in the plasma membrane of erythrocytes that pump calcium out of the cell into the interstitial fluid. There's a special pump that we talk about a lot, so you need to be familiar with this one. This is the sodium-potassium pump. It's a special dual pump required to maintain concentration gradients across the plasma membrane. What it does is it moves potassium into the cell against its gradient, right? So there's more potassium inside the cell than outside of the cell, while moving sodium out of the cell against its gradient. So there's more sodium outside of the cell than inside of the cell. This requires energy in the form of ATP, and it's continuously exporting 3 sodium out of the cell and moving 2 potassium into the cell. So what that looks like, here's step 1. You have your sodium-potassium pump here. It has 3 places for sodium to bind to and 2 places here for potassium to bind to. This is the cytosol, this is the interstitial fluid, and here's some potassium on the outside of the interstitial fluid. You can see it cannot bind to these sites yet. This protein or this pump needs to change shape for it to do so. So the first thing that happens, sodium ions bind, and then ATP binds, releasing energy. That energy causes a shape change or conformational change in the pump, and it opens up in such a way it releases the sodium ions, and the sodium can now travel out of the pump into the interstitial fluid against its gradient. ATP is broken down into ADP and inorganic phosphate in this process. Now you've got the 2 potassium ions that are able to bind to this protein. And when they do, the pump changes its shape back to its original conformation and releases the potassium inside of the cytosol, which is moving it against its gradient. Right? So normally there's a lot of potassium inside of the cell and not so much outside of the cell. And normally there's a lot of sodium outside of the cell and not so much inside of the cell. So what you've done is you've moved sodium from inside of the cell where it's a low concentration to outside of the cell where it's a higher concentration. So you've moved it against its concentration gradient. Same thing with potassium. You've moved it from an area of low concentration to an area of high concentration. So that's moving it against its gradient. All right, and then there's secondary active transport. This is also called co-transport or coupled transport. And this moves a substance against its concentration gradient. But it uses energy from movement of a second substance down its gradient. And so it uses the kinetic energy of that substance moving down its gradient to provide power to pump the other substance. It depends on the sodium-potassium pump for energy. They establish those gradients. And there's two types. There's a symport, where two substances are moved in the same direction, and there's an antiport, where two substances are moved in the opposite direction. So here's an example of a symporter. Here's the interstitial fluid. Here's the cytosol. And in the interstitial fluid, you'll notice that there's a high sodium concentration and a lower glucose concentration than inside of the cell. And what happens is glucose is transported up its gradient, but it's powered by this sodium diffusing down its gradient. And that basically provides the energy to allow the glucose to move through the symporter. This is an antiporter. You have a high sodium concentration in the interstitial fluid and a high hydrogen ion concentration in the interstitial fluid. And on the cytosol, it's lower. Both of them are lower. So what happens is sodium diffuses down its concentration gradient again into the cell, and this powers the pumping of the hydrogen ion up its gradient out of the cell. So it's going from an area of lower concentration to an area of higher concentration, and it's using the kinetic energy of sodium moving down its concentration gradient. Next, there's vesicular transport. This is an active process that's also called bulk transport. It requires ATP to transport large substances across the plasma membrane by means of a vesicle. And a vesicle is a membrane-bound sac filled with materials. There's two types of vesicular transport. There's exocytosis. This is large substances being secreted from the cell. For instance, macromolecules that are too large to be moved across the membrane. And then there's endocytosis. This is the cellular uptake of large substances from the external environment. You can go a step further and describe the three types of endocytosis, and you'll need to be familiar with all of these. There's phagocytosis, which is cellular eating. This occurs when a cell engulfs a large particle, and only a few cells in your body can perform this, for example, macrophages. There's pinocytosis, which is cellular drinking, and this is performed by most cells. And then there's receptor-mediated endocytosis. This uses receptors on the plasma membrane to bind molecules within interstitial fluid and bring those molecules into the cell. And again, this enables the cell to obtain bulk quantities of substances. Okay, here are examples of the three different kinds of endocytosis. So in this first picture, you have phagocytosis, or cellular eating. So you can see a particle is here against the plasma membrane. The plasma membrane invaginates and forms a vesicle around the particle. Pinocytosis, or cellular drinking, is very similar, except that instead of being a particle inside of the vesicle, it's a liquid. And here you have receptor-mediated endocytosis, where there's a ligand-receptor complex, right? So a ligand binds to a receptor. The receptor sends signals, and this recruits clathrin. And clathrin kind of coats this pit, this invagination, and causes the formation of a vesicle. I've shown this in this diagram as well. The particle being engulfed is, unfortunately for the cell, a virus particle, but you can see the virus comes into contact with the receptors. So this is receptor ligand binding. Signals are sent to recruit clathrin. The plasma membrane invaginates, and clathrin is coating this pit, and then it forms a vesicle, and after the vesicle is formed, clathrin kind of drifts off. This is a good video to watch for a summary of active transport. And just to go over this flow chart one more time, we talked about membrane transport. We talked about how there are passive processes that do not require cellular energy and substances move down their concentration gradient. And we talked about active processes, which require cellular energy and involves either the movement of a substance up its concentration gradient or it involves the formation or loss of a vesicle. Among the passive processes, you have diffusion and osmosis, diffusion being the movement of solutes, and osmosis being the movement of water. And below diffusion, you have simple diffusion or facilitated diffusion. Simple diffusion is where the particles, usually small and nonpolar, can directly cross the plasma membrane. And then you have facilitated diffusion, where a transport protein is required. And this can be either channel mediated, where an ion moves through a channel, or it can be carrier mediated, where a small polar molecule is moved by a carrier protein. Among the active processes, you have active transport or vesicular transport. In active transport, an ion or small molecule is moved against the concentration gradient. You've got primary active transport, which uses ATP for its energy, and you've got secondary active transport, which uses energy from the movement of a second substance. And that movement can be either in the same direction, so that would be a symporter, or it can be in the opposite direction, that could be an antiporter. In the vesicular transport column, you have exocytosis, which is release of vesicular contents from the cell. And you have endocytosis, where material is brought into the cell as a vesicle is formed. And remember, you have phagocytosis, which is cellular eating, where you bring in a large particle. You have penocytosis, which is cellular drinking, where you're bringing in extracellular fluid. Or you can have receptor-mediated endocytosis, where receptors are required. Okay, so questions. Which of the following does not rely on energy? Active transport? Well, no, because by definition, active transport relies on energy. Secondary active transport? No, that's a type of active transport. An antiporter relies on the energy of that second substance being moved down its concentration gradient, so that's not the correct answer. The correct answer is D, ion channel. Remember that these don't require energy. In fact, they help facilitate diffusion where no energy is required. Which of the following are formed when the plasma membrane surrounds a particle outside of the cell? The Golgi bodies? No. The rough endoplasmic reticulum? No. Secretory vesicles? No. Those are formed from the Golgi body. Or endocytic vesicles? Yes, that is the correct answer. Okay, so the resting membrane potential. This is an electrochemical gradient that exists across the plasma membrane. The plasma membrane establishes and maintains this electrochemical gradient, which is a combination of electrical gradient and concentration gradient across the membrane. So two components. Number one, the amounts of positive and negative charges are not equally distributed at the plasma membrane. So this is the electrical gradient. The inside of the cell, therefore, is more negative than the outside of the cell. You need to know that. And this is due to anions, or negative proteins in the cytosol, which are large proteins that cannot cross the membrane. So they stay trapped on the inside of the membrane. So that negative charge stays trapped on the inside of the cell. And it's also due to the action of the sodium-potassium pumps, where there's 3 sodium pumped out and 2 potassium pumped in. So there's an electrical difference there. And it's also due to the potassium leak channels. And then number 2, there's an unequal distribution of ions and molecules across the plasma membrane, and this is the chemical gradient. And you definitely need to know this. Potassium is the major cation inside the cell, while sodium is the major cation outside of the cell. Okay, so leak channels. The potassium leak channels are the single most important factor in setting the value of the resting membrane potential. So potassium efflux is potassium going from the cytosol to the interstitial fluid, and it leaves negative proteins behind. So you can see that down here. Here's potassium going from the cytosol to the interstitial fluid, and these large proteins stay trapped inside of the cell. The electrical gradient opposes the potassium efflux, but the chemical gradient facilitates potassium efflux. What does that mean? Well, that means that the electrical gradient, which you can see here, there are a lot of positively charged ions on the outside of the cell, and that would oppose a positively charged ion like potassium from leaving the cell. But there's much less potassium on the outside of the cell, and that facilitates the movement of potassium from the inside to the outside down its concentration gradient. If the potassium leak channels were the only channels open and present, the resting membrane potential would be negative 90 millivolts in a neuron. It's important to know that sodium diffusion also helps set the resting membrane potential. You can even see on this picture here that there's much fewer sodium leak channels than potassium channels. The electrical and chemical gradients both facilitate sodium influx, but only a relatively little sodium can flow in thanks to the limited leak channels. So it's more negative on the inside of the cell, and it's more positive on the outside of the cell. And sodium is a positive anion, so it wants to go from the outside to the inside. There's also less sodium on the inside of the cell than there is on the outside. So its chemical gradient is also pushing it towards the inside of the cell. But there's only a few sodium leak channels compared to potassium leak channels. So only a little bit of sodium leaks in, and its resting membrane potential, if there were only sodium leak channels, would be positive 20 millivolts in a neuron. So when you add this positive 20 and the negative 90, you get a resting membrane potential of negative 70 millivolts in neurons. Okay, so as far as maintaining a resting membrane potential, you need to know that the sodium-potassium pumps are significant. They maintain the potassium and sodium gradients following their diffusion, and there's 3 sodium pumped out and 2 potassium pumped in against their concentration gradients. Okay, let's talk a little bit about cell communication now. So we're going to start with ligand receptor signaling. Most cell communication occurs through ligands, which are molecules that bind with receptors. An example of ligands would be neurotransmitters and hormones. And the signaling is important for controlling growth, reproduction, and other cellular processes. There are 3 types of receptors that bind ligands. There's the channel-linked receptors, there are enzymatic receptors, and there are G-protein coupled receptors. So the channel-linked receptors, also called ligand-gated ion channels, allow ion movement into or out of the cells. And this occurs in response to a ligand such as a neurotransmitter binding. This helps initiate electrical changes to the resting membrane potential in muscle and nerve cells. And we'll learn about this more when we get to those sections. But you can see here, here's the ligand-gated ion channel. So here's the little gate, and it's closed right now. And then a ligand binds to the receptor, and the gate opens, and now ions are free to move into the cell. You also have enzymatic receptors. This is a receptor linked to an enzyme. You can see here, here's the receptor, here's the enzyme that it's linked to, and here's the ligand. So the ligand binds and activates the enzyme through the receptor. So now this enzyme is activated. And these are usually protein kinase enzymes. So what they go to do is what kinases do, and they go on to add phosphate groups to other enzymes within the cell. Okay, the third type of receptor to be aware of is the G protein coupled receptor. So what this does is indirectly activates protein kinase enzymes through a G protein. And you can see that here. So a ligand binds to the receptor. The receptor is coupled with a G protein. So when the ligand binds, it activates the G protein. The G protein goes on to either activate an effector protein, such as an ion channel, or it goes on to activate an enzyme, and the enzyme and protein channel send secondary messengers. So this is how it's indirect, right? So now there's a secondary messenger, and it goes on to activate protein kinase enzymes, which goes on to phosphorylate other enzymes, and the pathway continues until the desired result is achieved. So really what you need to know about G protein coupled receptors is that they indirectly activate proteins, enzymes, through a G protein. All right, DNA, chromatin, and chromosomes. So DNA is found in the nucleus and houses most genetic material of a cell. Each deoxyribonucleotide is composed of a 5-carbon sugar, which is deoxyribose, a phosphate, and one of 4 nitrogenous bases. There's adenine and guanine, which are the purines, and cytosine and thymine, which are the pyrimidines, and you need to know these. Each DNA has 2 complementary strands of nucleotides that form a spiral ladder. DNA is organized into units called genes, and when not dividing, DNA is in the chromatin form. So when it's dividing, DNA chromatin becomes tightly coiled into a mass called a chromosome. But when it's not dividing, that DNA is in the looser form, the chromatin form, that allows its genes to be read. And here's just an overview of the structure of DNA. So starting from the very bottom, you have the sides of the ladder, right? These are nucleotides. And here you have the nitrogenous bases in the middle of the ladder, creating the rungs. And you need to know which one bonds, hydrogen pairs bonds to which one. So you need to know that guanine pairs with cytosine and adenine pairs with thymine. All right, so we've got our double helix, and it gets wound around these proteins called histones, and that helps it kind of supercoil on itself and create a chromosome. So genes are segments of the DNA molecule, or segments of nucleotides specifically, that provide instructions for the synthesis of specific proteins. Now, remember that only 1% of DNA does this. About 99% of DNA is non-coding, means that it doesn't provide instructions for the coding of a protein. Each gene has a promoter region, which is the start signal, and a terminator region, which is the stop signal. Okay, so what is not true about RNA? It contains a sugar called ribose. That's true. Thymine is not one of its base pairings. That's true. RNA uses uracil instead of thymine. It is unstable in alkaline conditions. That is also true. Its sugar is more reduced than DNA's sugar. That is not true, because DNA's sugar is deoxyribose, right? So it's lost in oxygen and gained electrons. Therefore, it is more reduced than RNA. So D is the correct answer. Okay, what type of bond between complementary bases of DNA stabilizes the double helix structure? So if we go back to this image here, you can see these little dots between the nitrogenous bases represent hydrogen bonds. Okay, so you'll notice here between adenine and thymine there are only 2 hydrogen bonds, whereas between cytosine and guanine there are 3 hydrogen bonds. This makes the bond between cytosine and guanine a little bit stronger. And let's say that you had a segment of DNA that had primarily cytosine and guanine in it. That segment of DNA would be harder to break apart than a segment of DNA with, say, just adenine and thymine units. So the correct answer is C, hydrogen bonds. Which of the following statements best defines an organelle? Is it a layer of polysaccharides outside the plasma membrane of cells? No. Abnormal infectious proteins? No. Specialized structures within a cell? Yes. And collection of tissues used to serve a specific function? No, that would be an organ. Okay, so let's talk a little bit about protein synthesis, the process whereby cells make their proteins. This consists of 2 processes. First is transcription and second is translation. So transcription occurs in the nucleus. You need to know that. DNA is transcribed into mRNA. So think about this. What is transcription in general? It's where you copy something from the same language, where you transcribe it, right? So the cell is using the same language here. You're going from a DNA to an RNA. Both of those are nucleic acids. So it's transcription. Then you get to translation. This occurs on the ribosomes. The mRNA is translated into proteins. So this is a different language, right? You're going from a nucleic acid to a protein, so you're translating. There are 3 general types of RNA we'll talk about. Messenger RNA is the first, mRNA. Contains the code for the synthesis of specific proteins. So also know that the initial mRNA that's transcribed in the nucleus before modifications are added to allow it to leave the nucleus is known as pre-mRNA. So really you start off with transcribing a pre-RNA strand, and then that pre-RNA gets a bunch of modifications added to it that allow it to slip through those nuclear pores and leave the nucleus. Next you have transfer RNA or tRNA. This decodes the genetic message that's carried in the mRNA, and it carries amino acids over to be made into protein. Then there's rRNA, ribosomal DNA. This forms the protein synthesizing machinery of the cell. So ribosomal RNA helps form ribosomes. In fact, a ribosome is about 60% rRNA and 40% proteins. DNA directs the protein synthesis. We said there's transcription, which forms an RNA copy of a gene from DNA in the nucleus. And there are 3 events of transcription. There's initiation, elongation, and termination. And then there is number 2, translation, where the RNA is synthesized into a protein by ribosomes in the cytosol. So let's start with transcription. There's initiation. This is where the DNA is unwound by enzymes to make it accessible to RNA polymerase. And RNA polymerase is the enzyme that catalyzes synthesis of mRNA. So RNA polymerase attaches to DNA and locates the promoter region, and this serves as the start point for gene transcription. The bonds between DNA, those hydrogen bonds, are then broken and that region expands, and you can see that here. Here's RNA polymerase. It is bound to the DNA molecule and it has opened up the DNA strands so that they can be read and turn into mRNA. Next is elongation. This is where the pre-mRNA is formed using DNA as the template. So let's say that you had DNA and it had this as the template, the gene that you're trying to transcribe. So you've got cytosine, cytosine, adenine, thymine, guanine, adenine, adenine, cytosine, thymine. Well, your RNA transcript is going to be complementary to that, right? So where you've got cytosine, you're going to have guanine, and where you have adenine, you're going to have uracil, right? Because we're working in RNA. So the RNA polymerase just moves down the DNA and continues until the entire gene is transcribed. Then it terminates. The RNA polymerase is released at the terminal region of the gene. Hydrogen bonds that are broken between DNA and the new mRNA strand, and then the DNA rewinds back into its double helix. And you can see that here we talked about initiation. DNA is unwound by enzymes, exposes a segment of a gene. Here's RNA polymerase. It attaches to the promoter region of the gene, and it's going to read this DNA, this gene, until it finds the termination region. Next, you have elongation. RNA polymerase assists with complementary base pairing of free ribonucleotides. So you have free ribonucleotides out here just floating around, and RNA polymerase essentially grabs these and puts them in the order that they're supposed to be in. Finally, you have termination, where the RNA polymerase reaches the terminal region of the gene, and it's released. And then here's your newly formed RNA strand, which is released from the DNA strand, and then the DNA rewinds itself back into a double helix. And here's your pre-mRNA strand here. Okay, so we'll talk a little bit about modifications of mRNA. It's initially synthesized, it's more specifically called pre-mRNA, right after it's initially synthesized, and it undergoes changes before leaving the nucleus. Formation of mature mRNA requires splicing. So mRNA transcript has introns and exons. So introns are non-coding regions, whereas exons are the coding regions. So the exons, essentially, you want this gene to be full of exons, right? So here is showing exon here, intron here, exon here, intron here, and what happens is these introns get spliced out so that the mRNA strand is all exons. And those code for the protein that you're trying to make. And then once this occurs, it's now a mature mRNA molecule, and it's going to be able to exit the nucleus and enter the cytosol where the ribosomes are, so it can be translated into a protein. So this is a video going over transcription. You can watch it in your own time. Hopefully it's useful to you. Next, we're going to talk about translation. This is the synthesis of a new protein, and it occurs at the ribosomes, which are in the cytoplasm. mRNA is threaded through the ribosome. The ribosome acts as kind of like a table. mRNA is then translated into amino acids to produce a protein. This requires ribosomes, mRNA, tRNA, and amino acids. So ribosomes, or rRNA, they serve as catalysts during the assembly of amino acids into a protein molecule. mRNA is transcribed in the nucleus, like we just learned, from DNA, and it carries the instructions for synthesizing proteins. So mRNA is going to be a linear sequence of nucleotides. And you're going to read these 3 bases at a time. So this is called a codon, which is a 3-base unit. So this codon here, AUG, that's the first codon in this sequence. Then you have UAU, this is the second codon, and so on. There are a couple of types of codons you need to be familiar with. Most importantly is the start codon, which is AUG, and that's the signal to begin protein synthesis. And then you have the stop codons, and this is where mRNA reading ends, and I'll show you that here. Over in this column, you have AUG, which is the start codon, and it codes for methionine, an amino acid. Here you have your 3 stop codons, and I would be familiar with these. And you'll note that they do not code for an amino acid. All these other codons code for an amino acid. A little thing about this table, let's take this first section here. You have UUU and UUC, and both of these codons code for phenylalanine. You come over here to this column, you'll see that all of these code for serine. You've got UCU, UCC, UCA, and UCG. So really the only difference among these codons is the last nucleotide in the sequence. And this is called the wobble position, the 3rd position in the sequence, because a lot of times this nucleotide may change, but it still codes for the same amino acid. And that's redundancy that's built into this amino acid code here. And so the codons are said to be redundant or degenerate. As far as what you need to be familiar with in this table, you should definitely know the start codon and what it codes for. So start codon is AUG, and it codes for methionine, and that's always going to be the first amino acid in the protein when it's being made. And you need to know, or at least be familiar with, these stop codons, and know that they do not code for a specific amino acid. So tRNA, what it does is it brings specific amino acids to a specific mRNA codon. tRNA is cloverleaf shape, and it contains an anticodon region. So it's the anticodon sequence that determines the specific amino acid to which tRNA attaches. I'll show you what that means. Here's the cloverleaf shape of the tRNA. This is the anticodon region. And this binds with its matching codon, and you'll see that the anticodon determines which amino acid is attached to it, to the tRNA molecule. This is catalyzed by aminoacyl tRNA synthetase. You probably don't need to know this off the top of your head. But know that every amino acid has its specific enzyme, right? This is an enzyme that essentially helps attach its specific amino acid to it. And tRNA binds its specific amino acid prior to beginning of translation. So these guys are kind of out there already in the cytosol with these amino acids attached to them. And when it has an amino acid attached to tRNA, the tRNA is now called charged. Let's see here. Here's the messenger RNA. Here is the start codon, right? So that's going to code for methionine. Then you have a codon here, a codon here, and a codon here. And eventually you're going to get to a stop codon, and that does not code for an amino acid. Over here you have the ribosome. You have the large subunit up top and the small subunit down low. And it has these specific binding sites that the growing amino acid chain or growing protein is going to attach to. Okay, so this image walks you through the steps in the process of translation. So here we start with a tRNA that's carrying a methionine, right? Since it says the first amino acid, that's always going to be methionine. Here's the anticodon region, and here's the amino acid connected to the tRNA molecule. Then you have the large and small ribosomal subunits that come together and they form the ribosome. You have the mRNA molecule that kind of lays across the ribosome like it's a table. And the first step, just like in transcription, we have initiation. So here the components of the translational apparatus come together. So, and then you've got the mRNA laying on it, and here's the first codon of the mRNA. So this is the start codon. So the tRNA comes into this middle site here and binds and has the first amino acid hanging off of it. Next step is elongation, and during elongation, amino acids are brought to the mRNA by tRNAs and are added one by one to a growing polypeptide chain. So here's the second, here's another tRNA molecule that comes in. Sorry, that's just my dog Jim. Second tRNA molecule comes in, binds here, and the amino acids form a bond. Okay, so the amino acids form a peptide bond, and the mRNA continues to be read until it reaches a stop codon, and this is step 3, termination. And the stop codon, remember that the tRNA molecule associated with it is actually a release factor. It does not contain an amino acid, and so what this does when it binds here, it releases the polypeptide strand, and you have a completed polypeptide or a completed protein. This is another video you can watch on your own time. Hopefully it'll be useful. So in summary, transcription and translation. You've got transcription, which occurs in the nucleus. Pre-RNA is transcribed from DNA, and then it's processed to form mRNA, and then it leaves the nucleus. When it leaves the nucleus, it goes to a ribosome, which ribosomes are in the endoplasmic reticulum and in the cytosol. So mRNA is read to direct tRNAs in adding amino acids, and then a protein molecule is formed. So questions. Transcription of the genetic material occurs when A, DNA is copied identically to be distributed to its daughter cells. B, DNA is copied into the complementary form of mRNA. C, DNA is converted to the coded protein. Or D, the message is edited into a form that the ribosome can understand. The correct answer here is B. DNA is copied into the complementary form of mRNA. Remember, transcription is when you go from one nucleic acid, DNA, to another nucleic acid, RNA. You're using the same language. Where does translation occur? A, in the nucleus. No, that's where transcription occurs. B, in the centrosome. No, we'll talk about the centrosomes in a little bit, but they're not involved in translation. C, in the ribosome. It's on the ribosome that it happens. And then D, in the cytoplasm. So I would go with D on this one. It occurs in the cytoplasm. It occurs on the ribosome. But this, I don't think, is the best question, as you could get these two confused, so I won't ask an ambiguous question like this on the test. And then what is the role of ribosomes? Is it to make proteins, for waste removal, for transport? And then I can't read this last one. But the role of ribosomes is A, to make proteins. Okay, next, we're going to talk about cell division. Cell division is where one cell divides to produce two cells. It's necessary for development, tissue growth, replacement of old cells, and tissue repair. So there's mitosis, which is cell division that occurs in somatic cells. And somatic cells are all the cells in your body other than the sex cells, so other than the ova and the sperm cells. And in mitosis, the cells that are formed, the daughter cells, contain 46 chromosomes, so that's the normal number of chromosomes. Meiosis, on the other hand, is cell division in sex cells, so these are the cells that give rise to sperm or oocytes. And their daughter cells contain half the number of chromosomes as a somatic cell. And that's because when two sex cells form, they join their chromosomes together and have 46 chromosomes altogether. To talk about cell division, you have to talk about the cell cycle. What occurs in the cell cycle is replication of cell components. So then the cell will divide into two identical cells called the daughter cells. And there's two major phases in the cell cycle. There's interphase, which is this whole blue area, which lasts approximately 23 hours in most mammal cells. And this is where the replication of cell components occurs. And then you've got the mitotic phase, which in most cells lasts about 1 hour. And the mitotic phase is where the cell actually, the nucleus and the cell itself split into two. So interphase, this is the time that the cell prepares for division. The cell is making a bunch of enzymes and proteins, and then in the S phase, it makes DNA. So this is all in preparation for division. DNA is in loosely coiled chromatin form in the interphase. This allows it to be read and genes to be expressed. There are three phases of interphase. There's the G1, the S, and the G2 phase. So we'll start with the G1 phase. This is the first gap stage. This is where growth and production of new organelles occur. The structures that are needed for DNA replication are formed. The replication of centrioles to produce two pairs of centrioles forms. And then in this phase, the cells are diploid, meaning that they contain one pair of each chromosome, one maternal copy and one paternal copy. So cells in this stage have 46 chromosomes, 23 from mom and 23 from dad. Next is the S phase, and S stands for synthesis, that is DNA replication. So here, DNA is replicated. It's going to require deoxyribonucleotides, right? Those are the building blocks of DNA. And it's going to require an enzyme called DNA polymerase, and that just reads the DNA and produces a new DNA strand. This occurs in the nucleoplasm of the nucleus. Cells in this stage go from 2N to 4N. So now you've got two maternal copies and two paternal copies of the chromosomes, and you can see that here where you've got a maternal copy and a paternal copy. You've got 2N, right? So imagine each one of these chromosomes is N. It replicates, and you get now what are called sister chromatids. So now you have two sister chromatids from mother and two sister chromatids from father, and altogether that's 4N. And then when the cell goes to divide, one copy from mom and one copy from dad go into each cell, and you're back to 2N. As far as the steps of DNA replication, this is going to look familiar to you. It's similar to transcription. So there's going to be unwinding of the DNA molecule. You're going to break those parent strands apart. So you're going to break those hydrogen bonds that are holding the DNA molecule together. You're going to assemble a new DNA strand or strands, and then the DNA helix is going to wind itself back. The DNA is going to wind itself back into a double helix. This process continues until both strands are replicated, and these replicated strands are now referred to as sister chromatids. And they're attached in the middle, which is not pictured here, but in the middle there's an area to which they attach, and that's called the centromere. Okay, so here are the steps in DNA replication. The first step is going to be unwinding of the DNA molecule, and there are special enzymes that do this. 2, the breaking of the hydrogen bonds between bases of the two DNA strands occurs. And then you're going to have DNA polymerase come in here, and it's going to read the DNA, and it's going to form new strands complementary to the strand that it's reading. Then when it's done, it releases the complementary strand or the replicated strand of DNA, and the original DNA restores itself into the double helix. Then next is the G2 phase, the second gap phase. This is the last part of interphase. It's very brief, and essentially you're still producing enzymes and other proteins that you need for division. And the stuff that got started in the G1 phase, like centriole replication, that's completed. Everything gets completed right before mitosis. So in summary, you have the G1 phase where the cell produces necessary molecules needed for DNA replication. The cell grows and reproduces organs, and it's the beginning of centriole replication. Then you have S phase. This is where DNA gets replicated. And then G2 phase, the centriole replication and the other structures needed for cell division get completed. All right, and then there's the M phase, or the mitotic phase. This follows interphase, and two events occur in it here. You have mitosis, which is the division of the nucleus, and then you have cytokinesis, which is division of the cytoplasm. And there are four phases of mitosis. There's prophase, metaphase, anaphase, and telophase. So prophase is the first stage of mitosis. The chromosomes appear due to coiling of chromatin. The nucleolus is broken down, and spindle fibers, which are made of microtubules, grow from the centrioles. The centriole pairs start to move to opposite poles of the cell, and the nuclear envelope breaks down. So here this is showing the nucleus. The nuclear envelope is starting to break down. You've got these dark sections here, which are the chromosomes starting to form. You have these fibers radiating out from each pole of the nucleus. And these are spindle fibers. They'll come into play when the cell divides. And the nucleolus is broken down. Next is metaphase, the second stage of mitosis, where the chromosomes are aligned on an equatorial plate of the cell. And then the spindle fibers extend from the centriole and attach at the centromere of each chromosome. So you have here what was formerly the nucleus, right? You've got these chromosomes lined up in like a perfect line across this area. And you've got spindle fibers reaching from the centriole, attaching to the middle of these chromosomes at their centromeres. And what's going to happen is these spindle fibers are going to pull these chromosomes apart in opposite directions. And you see that happening in anaphase. So starts as the spindle fibers move and the sister chromatids are pulled apart. Each chromatid is now called a chromosome, and it's a chromosome of one DNA double helix with its own centromere. And last, you have telophase. This is the arrival of each group of chromosomes at each pole. The chromosomes start to uncoil and begin to return to chromatin. New nucleoli are formed in each cell, and a new nuclear envelope forms around the chromosomes. So this is the end of nuclear division, is telophase. And then very last, you have cytokinesis, which is the other major event in the mitotic phase. And this is where the cytoplasm divides evenly between the two newly formed daughter cells. And it tends to overlap some with anaphase and telophase. You'll see this here, the cell membranes kind of start to pinch down, and it pinches off, and you've got two new daughter cells. Here's a video that you can watch covering the cell cycle. And then here's just an overview, right? We talked about G1, S, and G2 phase. So the big picture here is what's happening in interphase is the cell is preparing for division. So the cell is making new enzymes, new proteins, and it's replicating its DNA. And then you've got mitosis, in which chromosomes form, are lined up, then are pulled apart, and then the nucleus starts to close up around each set of chromosomes, and then finally the cell itself splits into two. So what type of cells result from mitosis? Is it one diploid cell, two haploid cells, two diploid cells, or four haploid cells? Well, we know right off the bat that A and D are wrong because 2 cells are formed from mitosis. So it has to be either B or C. And we know that the answer is C, 2 diploid cells, because remember, if we go back, if I can find it, we talked about here, where the cells are 2N at the beginning, then they go to 4N during replication of DNA, and then at the end of mitosis, you're going to have 2 daughter cells with diploidy, which means that there's 2 copies of each chromosome, or 2N, one from mother and one from father. Which of these does not match the nitrogenous base to the nucleobase? So is adenine a purine or a pyrimidine? Adenine is a purine, so A is correct. Is thymine a purine? No, actually, thymine is a pyrimidine. So is uracil, and so is cytosine. So you need to know which bases are the purines and which bases are the adenines. And the correct answer here is B, purine and thymine, because thymine is actually a pyrimidine. In which phase of the cell cycle does DNA undergo replication? Very important to know that it is the S cycle of interphase in which DNA undergoes replication. Okay, so a little bit about cell aging and cell death. So aging is a normal continuous process with obvious body signs. When you become older, you have a lower number of normally functioning cells, and you have some abnormal function of the remaining cells. You can have alternation in structure or number of organelles, so you can have fewer organelles than normal, or the structure of the organelles can be abnormal, and so they don't work as efficiently. You also get changes in the structure of chromatin and chromosomes as you age. And then cells die in 2 ways. They're either killed by damage or harmful agents like pathogens or UV radiation, or they go through what's called apoptosis, and that's programmed cell death. And this occurs to remove harmful cells and promote proper development. So, and let's say you are a respiratory cell that gets infected with a virus. You may signal to yourself to undergo programmed cell death, and then you would essentially release your lysosomes and digest yourself. Another reason that apoptosis occurs is during fetal development. So for example, the fingers, when the fingers are formed, there are regions of tissue or webbing between the fingers, and so those cells in those tissues undergo programmed cell death or apoptosis and empty out that space between your fingers.